You ever think about the epic life story playing out in, say, a single drop of water? Well, today we're going to dive right into that microscopic drama. It's the story of a bacterial colony, a tale of explosive growth, intense competition, and eventually decline.
It sounds a little crazy, right? I mean, how can one single invisible cell create billions of copies of itself in less time than it takes to get through a workday? The secret lies in a method of multiplication that is brutally simple and unbelievably powerful. So, meet our main character. This little bacterium doesn't need a partner. It is a master of self-replication. It uses this incredibly efficient process called binary fission, and that is the key to its rapid takeover of the world. All right, let's start the story at the very beginning. That first crucial split. How does one cell manage to become two perfect, identical copies of itself? What's wild is just how straightforward this whole thing is. First, the cell makes a perfect copy of its DNA. Then it starts to stretch out. A new wall, called a septum, forms right down the middle, and then, poof, it pinches apart into two identical daughter cells. It's basically cellular cloning happening again and again and again. And the little molecular machine that drives this final split, it's a protein called FITZ. Just imagine a tiny belt wrapping itself around the cell's middle. This Z-ring, as it's called, just cinches tighter and tighter until it neatly pinches the parent cell in two. It's the little engine that could for bacterial reproduction. Okay, so that first split, that's just the beginning. Let's jump to the next phase and see how this one simple act of division leads to a mind-boggling population boom. And right here, you can really see the insane power of doubling. 1 becomes 2, 2 become 4, 4 become 8. It doesn't sound like a lot at first, does it? But this is exponential growth, which means the population doesn't just grow, it absolutely explodes. The key ingredient here is speed. How fast can this doubling happen? We call this the generation time. And let me tell you, it varies like crazy. A super common bacterium like E. coli can double every 20 minutes. But the one that causes leprosy, it takes a leisurely 14 times days. In the microbial world, speed is absolutely everything. So let's try to wrap our heads around this. Imagine you start with a pretty solid population, 100,000 cells, and their generation time is a pretty average 30 minutes. What do you think that colony looks like after just 2 hours? Go on, take a guess. In only 2 hours, that's 4 doublings. You go from 100,000 cells to, get this, 1.6 million. That's a 16-fold increase in the time it takes you to watch a movie. The power of exponential growth is it's staggering. But look, a party this big can't go on forever. Just like any great empire in history, a bacterial colony has a really predictable life story. A dramatic rise and, you guessed it, an inevitable fall. And this is the roadmap for that story. It's called the bacterial growth curve. Think of it like a 4-act play, and pretty much every bacterial colony in a closed system follows this exact same script. So let's break it down act by act. Act 1 is the lag phase. So the bacteria have just shown up to the party, and the numbers aren't increasing yet. They're busy, checking out the new plays, adapting, and getting all their gear ready for the massive expansion that's about to happen. It's like an army gathering its resources before a huge battle. And then comes Act 2, the log phase. This is the main event. There are tons of nutrients, plenty of space, and the colony kicks into that explosive, exponential growth we were just talking about. The cells are at their absolute healthiest. And what's really interesting is that this is also when they're most vulnerable to antibiotics, because those drugs specifically target these fast-growing processes. But then we hit Act 3, this stationary phase. The party's starting to wind down. The colony's own incredible success becomes its biggest problem. The food is running out and toxic waste is piling up. The growth rate grinds to a halt until it's the same as the death rate. The population hits a ceiling and everybody switches into a desperate survival mode. And that brings us to the final tragic conclusion, Act 4, the death phase. The environment has just become too toxic and depleted. Cells are now dying off way faster than they can reproduce, and the total number of living cells just plummets. The once thriving city collapses. So we know this whole epic drama is unfolding, but how in the world do scientists actually watch it happen? How do you even begin to count a population that is so massive and yet completely invisible to the naked eye? Well, scientists have two main ways of getting a head count. The first is a direct count. Think of it like a total census. It counts every single cell, whether it's alive or dead. But a viable count is different. It only measures the living cells, the ones that can still reproduce. That tells you the size of the active army.
Okay, but if you're trying to do one of those viable counts when the colony is at its peak, you've got a huge problem. There are millions, maybe even billions of cells. You can't possibly count all of them. So you have to do something else first. You have to dilute them.
And the solution is this really clever technique called serial dilution. You take a tiny bit of your sample and you dilute it, usually by a factor of 10. Then you take a bit of that new sample and dilute it again, and so on. If you try to grow the early dilutions, you just get a smear. It's too numerous to count. But eventually, you get a plate with a nice, countable number of colonies, where each little dot grew from just one single living cell.
So there you have it, from one cell to billions and then all the way back to collapse. This predictable cycle, this boom and bust driven by simple division and limited resources, isn't just a story happening in a lab dish. It's a fundamental pattern of life. It really makes you wonder, what can this tiny, invisible drama teach us about growth, resources, and the limits of any environment we live in?